Hello, APBIO. This is the second of your immune system videos. Oops. So I just want to just go over just quickly where we were. So we were over here in your innate immune responses, and this is a great overview diagram. You can spend some time looking at it. This is showing you where we talked about macrophages, and we didn't talk about immature dendritic cells. But they are also cells that are going to be able to gobble up pathogens over here, non-specific cells. They're going to gobble up pathogens. This next step that we're going to talk about is sort of bridging the gap between these inborn responses, so non-specific responses to pathogens, and then shifting over and stimulating your adaptive response. So your experience-based response to pathogens. It's called your specific. So this is non-specific. It's anything that's not you. And now this is specific pathogens. So we're going to talk about this guy right here. How these guys are actually converted to this guy. So let's go in your notes back to where we were. We're going to talk about this process of, or this next type of protective cell. It's another white blood cell. It's a group of white blood cells that actually can be converted to antigen presenting cells, and they're professional antigen presenting cells. The reason we call them professional is because you have all of your cells have the MHC1 on the outside, and they can all present a distress flag and say, please, natural killer cells, come and get me. I'm having a problem. Or even they tell any cells that they're having a problem. And all these, all the white blood cells that are phagocytic will come along and eat any cell that's in distress. That's recognized as a pathogen. A cell in distress is recognized as a pathogen. But anything with MHC1 is, you can think of as an amateur antigen presenting cells. The professionals have major histocompatibility too. So it's a slightly different protein, and it's also a cell surface protein, and it's on the cell surface of these professional antigen presenting cells. So they're the group of immune cells, and their job is to engulf pathogens, and then they're going to present a piece, or just, you can think of it as displaying a portion of that pathogen on their surface. So let's see how that would happen. So this is a dendritic cell or a macrophage, a white blood cell, a non-specific white blood cell that's going to eat a pathogen. This little thing right here is a pathogen. I should have labeled it in my diagram. And then you can see here, now it engulfed it by phagocytosis. So this is either a macrophage or a dendritic cell. You can see it here. That's what they're showing in step one. So this guy is non-specific, and he ate one of these, and now it's here. Now what he does with that, this cell right here does with that is it's inside this food vacuole. I think it's called a phagosome. And it fuses with, the garbage dump guys, lysosomes, and then the lysosomes break it up, and then it takes it and holds it on its surface here with that MHC2. And when it does that, it's matured into an antigen presenting cell. That's what APC means. And now it can it has information about a specific pathogen. So this really is sort of the messenger in between. Here's our antigen presenting cell. It's holding that fragment of the pathogen on the outside. Now it can go ahead and stimulate your specific immunity, your innate immunity. So it's passing this info along, and it acts as a messenger in between your innate and then these acquired protective systems. So now we're going to move over to the acquired system. The first cell that's going to be activated is a T cell, and specifically it's a helper T cell. So let's look and see where that is over here. This is a helper. This is showing a secondary response. Oh, I don't want to show this video. So and a helper T cell. Let's go over to your acquired immune system responses. Let's go here first. Over in your acquired, or it's also called your adaptive immunity, these are all based on experience. So this is the experience that it's talking about are the information from specific pathogens. And you basically have two systems. You have a system that's protecting your body fluids. That's your lymph and your blood. And but it's protecting you against pathogens. So this system in your body recognizes pathogens, period. But the pathogens can also enter into your cells. And so you have a system and white blood cells that are specific for targeting your own cells that have been quote unquote breached. So this is the last ditch effort once your cells have actually been infected. So let's talk about this humoral response first. In general, the way that your acquired system works is, first of all, they have, again, we're talking about the two different cells that are going to be active in your acquired system. They're both leukocytes, so they're both white blood cells, but these are not phagocytes. They're your innate, they're your specific white blood cells. They're called lymphocytes, and there's two types. There's B cells and T cells. And they're going to be activated by a pathogen, but the pathogens, when we talk about them in your specific defenses, in your adaptive immunity, we call them antigens. And an antigen is just a pathogen that results in a specific immune system response. And it's called an antigen because it's an antibody generator, antigen. And this is actually the branch of your immune system that vaccines take advantage of. Vaccines are just intentionally stimulating your specific immune system response. You're given an antigen, and then your body launches this crazy attack to protect you from the antigen. Okay, so let's talk about this defense of your fluids. It's called a humoral response. You're just defending your body fluids. These are pathogens or antigens, essentially, that are in that are outside cells but inside your body. And if we look at what that pathway looks like, here's the antigen right here, and a macrophage, which is part of your innate system, is going to come along and eat this, and then it's going to display a portion of it using MHC2 on its surface, and it's going to take this information, this specific information from this specific pathway, and it's going to activate a helper T cell, but it's going to pass along that specific information. So now this helper T cell that was before a naive helper T cell, it just was a T cell that was floating around that was non-specific, has now been specified and imprinted with this particular information from this particular pathogen that this particular antigen presenting cell is given it. Now, there's two things that this helper T cell is going to turn on, two different pathways. One pathway to protect infected cells. We're not going to talk about that one right now. We're going to talk about this pathway to directly interact with body fluids. And what it's going to do is activate what's called a B cell. So a B cell is a white blood cell, and specifically it's a lymphocyte. So it's a type of leukocyte, specifically a lymphocyte, and it's got all these all these antibodies on the outside of it. And its job is actually to produce antibodies. And each B cell that's been activated by a T cell, there are naive B cells as well, which are B cells that haven't been specified yet. And they're waiting to be specified. And the specification conversation is this one that happens right here. So an activated helper T cell is going to activate this B cell. And what that means, it's been, you can think of it as being imprinted with a specific pathogen. So like I said, on the outside of these, there's all these Y-shaped proteins. Those Y-shaped proteins are actually antibodies. So antibodies can exist in two different ways. And every antibody in your body is specific for a specific pathogen. And the whole Y thing is all the same, except that arms are what are variable. And what they do is they are going to a ton of the antibodies are produced, and they also exist on the outside of these B cells. So they're in two different places. They're attached to cell surfaces, and then they also float freely in your blood and in your lymph. Again, just clarify that they're a protein, they're not a cell, but they are going to swarm a pathogen. They're going to interact directly with pathogens. And then they're going to immobilize the pathogen and signal using cytokines and using interleukins to come over. They're calling other immune system cells to come in and deal with the situation. I got a situation here. Yo, got a situation. So they're really a signaling. They're going to launch a signaling cascade that's called the complement system, and you get all of these different proteins and disease fighting pathways that all start. The clumping process that's caused by antibodies and B cells is called agglutination. And that's just the clumping of the pathogens with these proteins. Now, what happens when the B cell, the naive B cell, is activated? It goes through a process called clonal selection. And clonal selection refers to the really rapid reproduction, the preparation to fight. So here's this B cell, and it's been activated now that matures in the bone marrow. It's been activated by a particular helper T cell. Now it's specific for a pathogen, and it's going to go really, really fast, produce all of these different cells. So all of the exact same cells, sorry. That's why it's called clonal selection. And it's going to actually produce two different types of B cells. Most of them are going to be what are called effector B cells. And then it's going to produce some memory B cells. But these are also known as plasma B cells, and they're active fighters. So they're going to go around, and they have these Y-shaped proteins that are specific for a particular pathogen. And they're going to go around and find that pathogen and bind and call for help. They are really high in rough endoplasmic reticulum because their job is to pump out hundreds of antibodies. So they're secreting lots and lots of proteins in the form of these antibodies. Now, some of the cells that are produced by clonal selection are memory B cells, and these are going to store information from that specific pathogen to next time that you are presented with that same pathogen. The secondary response is so fast that the infection's not successful. So let's see why that's the case. Let's talk a little bit about that. This process right here, from antigen all the way to antibodies, takes a couple of days to happen. So in the meantime, whatever the pathogen is, it's had time to grow in numbers as well. I mean, think about how fast the generation cycle is of viruses, or the generation cycle of bacteria are. But in the secondary response, we're starting all the way down here. So all that has to happen is clonal selection and Shazam. So clonal selection is really fast, but none of this stuff has to happen. So this is less than 24 hours here. This is hours, and this is days. So in these days, the pathogen has time to launch an attack. Here, no way. We are way faster than that pathogen is able to launch an attack, and before it even can gain any speed, we smosh it down. And that's why you don't get the same thing twice. It's not that you can't be quote unquote breached by the same substance twice. It's just that your memory cells have the ability to fight that pathogen that much more quickly. Now, this is fighting the humors or the fluids of your body, but now I'm going to talk about in the next video how you protect your cells that have already been infected. Okay, I hope that was helpful.